Good afternoon, everybody. Thank you for your presence here in the last session of this meeting. I hope it will be helpful and interesting for all of you. We'll try to keep on time, and I'll make just a brief presentation. I don't think that cancer, that's the topic we are dealing with today, is something you need to be very explain how it is and how important it is. Just to remind that's a very much a large cause of death all over the world. There are many differences from the clinical, economic, risk factors and other aspects. And it's expecting that in the few next years, its incidence will go increasing due to several factors such as population getting older, pollution, and some other factors. So that's the reason why prevention, early diagnosis, and equity and affordability of the treatments are so important aspects and so important issues. We have here three experts on those fields, and I'm sure that they will clarify some aspects and make some interesting apportations. Without further delay, I think we will start with our first speaker. That's Dr. Barbacid. It's also well known to all of you, I imagine. It's a distinguished, international recognized molecular biologist with significant, very important contributions to the molecular biology of several tumors, discovering some oncogenes, dealing with some other molecular importance in the cell cyclones as cyclins. He has a large number of publications. He has an H-index amazing. And I think that's quite a pleasure, quite an honor to have him here to talk to us about the first talk of this session. The title of the talk is The Cancer Cure Dilemma, Past and Future of the Precision Medicine, and I hope that it will be quite interesting. Please, Professor Barbacid.
Well, thanks very much for the introduction, and thank you for the invitation. Real pleasure to be here. So, for the sake of time, let's move into. Is there yeah. Okay. So, as we all know, the first quarter of the 21st century has been witnessed a true revolution in the generation of new therapeutic armamentarium against cancer, mainly precision medicine, immunotherapy, and CAR T cells. Notwithstanding their significant impact in the treatment of many cancers, these new therapeutic strategies have their own challenges that are limiting both their use and their efficacy. And obviously, precision medicine is not an exception. Let me just list quickly the main challenges of precision medicine. First of all is the number of targets. We already know more than 500 oncogenes and tumor suppressors identified in a variety of cancers. Okay? And industry, in spite of its power, has only been able to generate inhibitors or now more recently degraders against a few dozen targets. Okay? So less than 10% of the human oncogenes or cancer-causing genes now have a selective drug, okay? Less than 10%. So that's obviously is a main challenge. The other one is how to target tumor suppressors. As we all know, tumor suppressors cause cancer for not being there or for being inactive. So therefore, inhibiting them will not do anything. If anything, will make them worse. So making them active is a challenge. And for instance, for P53, they've been working at least for 40 years. I still remember the nutlins when I was in the United States. So and it hasn't worked, okay? So they have to target the signaling pathways, which is obviously an indirect way of targeting, and presumably is going to be less efficient. And we have the example, probably the best example of a good therapy will be the PARP inhibitors in people with BRCA mutations. Then the other one is selectivity. The similarity between the oncogenic PARP and their normal counterpart makes selectivity to be a real challenge, because it's very difficult to make a drug that only targets the mutant form. Even though it selectively targets the mutant form, still slightly that affects the normal counterpart. Another challenge is targeting strategies. Until now, most of the inhibitors in precision medicine are inhibitors of enzymes. But there are many oncogenic proteins that are not enzymes, and some of them we still don't know how pharmacologically target them. Okay, now the new chemistry, Degron chemistry, products, hopefully will resolve these problems. Then you only need to have a molecule that binds to an area. Doesn't have to be an active area, but still, it's not an easy task. It's a hope, but still there are very few drugs that degrade the target rather than inhibit the target. And last but not least, certainly, is tumor resistance. Tumor resistance, basically all precision medicine drugs have shown that when used as single drugs, as single therapy, they develop resistance very rapidly, limiting their efficacy. And since the Chinese say an image is worth 1,000 words, here you have a very well-known study published, I think it's now more than 10 years ago, in which they have a patient with multiple melanoma with a BRAF V600E mutation that responded very well to vemurafenib, a drug that actually still has one of the records of having been developed in the shortest period of time, just 8 years in a company called Plexicon. However, when this patient waited a little bit longer, time passed. Here it goes back to to where he was a year earlier. So the drug, the disease is now completely resistant to vemurafenib. So the question is, what can we do? Okay. One possibility is to use combination therapies. I always say that if somebody coming from Mars, from outer space, will come to Earth and tell them, say, well, the worst disease that we have is a disease that has 3,000 mutations, 30,000 mutations in the case of melanoma, or lung cancer, smokers, and we are so smart that we are going to cure it with only one drug, we'll think that we are crazy. I mean, you cannot cure a disease with 30,000 mutations with only one drug. So maybe combination therapies will be one way, at least, to solve the problem of tumor resistance. However, obviously, their efficacy, remember I told you that one of the challenges of precision medicine is the similarity between the normal protein and the mutant protein, the oncoprotein, is that the combination of toxicities of the 2, 3, 4, 5 different drugs in the combination therapy has to be well tolerated. Otherwise, it won't be able to use it in nature. So, in the clinic. So, what I'm now going to tell you in the second part of my talk is just a practical example. So, we are not just talking about hypotheses and theories. And the example is are the KRAS oncogenes. Why? Well, KRAS oncogene is one of the most frequently mutated oncogene in human cancer. It's present in almost a quarter of all human solid tumors, but not only is present in a quarter of solid tumors, it's present especially in the 3 main killers pancreatic cancer, colorectal cancer, and lung cancer in different proportions, but certainly it's almost a universal presence in pancreatic ductal adenocarcinoma. It makes it just, as you know, pancreatic ductal adenocarcinoma is the worst, the tumor with the worst survival. So, and incidence is increasing. So this is clearly targeting KRAS is a major medical need. So for those of you who are not familiar, I think it's with the KRAS oncogenes, I think it's good to learn how long things take in cancer and in oncology. KRAS was discovered originally in retroviruses, but as it relates to human disease, was discovered 1982. Its mechanism of activation by a single point mutation was also discovered that year. 2 years later, it was the activity of its gene product. It was found to be a small GTPase. Then the tertiary structure was resolved fairly soon. After all, it's a small protein, but that didn't help in terms of developing drugs. 10 years later, it was 1993, the signaling pathways were started to be uncovered. But it took more than 2 decades to identify an inhibitor. In fact, during a long time, more than 2 decades, KRAS was thought to be an undruggable molecule. Okay, so fortunately, through a good medicinal chemistry, it was found a small chemical inhibitor that then in 10 years, or actually less than that, in 8 years, led to the first commercial chemical FDA-approved inhibitor, a drug made by Amgen called sotorasib. But even the same year, the first evidence of rapid resistance appeared in a paper of the New England Journal. So, and in fact, a study led by a Spanish clinician, Luis Pezares, show, unfortunately, that the overall survival of pretreated cancer patients with sotorasib was not any better than with chemotherapy. Okay, even though it was better in the sense that there is less, that there are fewer side effects, but still overall survival is the same. And the results with pancreatic cancer were even worse because that very same year, in another paper published in the New England, the cancer patients, the pancreatic patients treated with sotorasib, their overall survival was less than 7 months. So clearly, after all 40 years, more than 40 years, trying to find inhibitors of KRAS, at the end of the day, now we have them, but they are not very useful. Okay, so this, I think, it summarizes or epitomizes the difficulties and the limitations of precision medicine. Clearly, I'm showing a difficult case. There are other cases where the results were a little bit more optimistic, but again, we have to keep in mind that less than 10% of the cancer targets have now selective drugs. So we have a lot of work to do for the next generations. So I'm just now going to finish by telling you very quickly some results in our own laboratory that are giving us a little bit of hope. Okay, well, this is a more recent this year. There were a couple of papers in Nature where they now are using pan-KRAS inhibitors because, well, I'm not going to get into the details. KRAS can be activated by different mutations. So these are new inhibitors that can target all the mutations, which is also an advance. But they live longer, but at the end of the experiment, all the mice are dead. So is there a solution? Then I come back to the combination therapies, okay? So we have hypothesized that perhaps the best way to prevent tumor resistance is to combine direct KRAS targeting with targeting KRAS signaling. Targeting KRAS signaling at independent nodes, either downstream, upstream, or even in orthogonal pathways. And again, the idea will be that if you're targeting not only the mutant protein, but the way it signals, perhaps the cells, the tumor cells, don't have enough time to rewire all their signaling, all the tumor selective signaling, in a way, conceptually, not too different in the way that HIV infections were conquered. The first drugs were where people were treated with only one drug, and they developed resistance very rapidly. And the only way that HIV, where infections were conquered, was through a combination of at least 3 drugs. So we used pan-KRAS inhibitors developed by a small biotech company in San Francisco, Revolution Medicines, and we use experimental pancreatic tumors that have been, described first by Dave Tuvison right after the beginning of the century, in 2001, actually, as a matter of fact. Now, this is the results, okay? So you have, in the vehicle, the tumors grow very fast. Now, when you use the pan-KRAS inhibitors, they don't grow that fast, but they still grow. In fact, if you use a Kaplan-Meier representation, the mice basically double their life, their survival. Okay, if we could do this in humans, won't be bad, but obviously we know that mouse data is not as good when it goes to humans. So clearly, this is not enough. What we've done is to combine inhibiting KRAS with the inhibition of 2 downstream targets, one upstream and one orthogonal target. And to our surprise, in a way, because we were not expecting this, but the tumors have disappeared, okay? And we have now mice living almost a year that are tumor-free, okay? So you can now use the Kaplan-Meier, and it's certainly much better. We have now, these mice are 150 days, and still they are tumor-free, okay? So, I remember that I told you one of the potential problems of combination therapy is the accumulation of toxicities, at least in mice. Unfortunately, mice are much harder than we are. So lack of toxicity in mice is not a guarantee. It's not going to be toxicity in humans, but at least it's a good sign. Obviously, it wouldn't be bad if the mice would die due to toxicity. The mice are perfectly fine. Their GI is fine. They don't lose weight. Now, obviously, we are doing a few more studies to try to see potential toxicities, but so far, no evidence of toxicity. So, and these are human tumors, put in mice, the famous PDX, patient-derived tumors, and as you can see, they also respond very well. So, to finish, in summary, treatment of experimental pancreatic tumors, as well as pancreatic tumors from patients, with KRAS inhibitors only generate modest increases in survival due to the rapid appearance of tumor resistance, one of the main challenges of precision medicine drugs. In our case, we've been able to eliminate these tumors and to make them disappear. They, as I said, 150 days in a mouse is a long time, okay? Mice live 2 years. So basically, this is equivalent to maybe about 15, 20 years in humans, assuming that we could reproduce this in humans, obviously. So the question here is open to the future as to whether combination strategies will work on cancer patients. Clearly remains to be, to be demonstrated. As my clinical colleagues tell me, my tumors are bigger than your mice. So we need to wait until we can get these inhibitors to be able to go into clinical trials and see whether we are lucky. We don't expect to have the same results as we get in mice, but even if we get them half as good, I think it will be a big advance. And especially this should be applicable to other therapies in precision medicine. Thank you very much for your attention.
Thank you very much, Dr. Barbacid. I don't know if there is any questions. We should have time for one short, rapid question, if you have. If no, I'd like to ask you just a comment on, do you perceive any of the targets that are available now as more promising than others for getting a cure of solid tumors?
Well, I mean, we limited what we can test. We have found this combination, it works. That doesn't mean that you cannot find a better combination for sure, or less toxic. So this is just a very, very beginning. This is not even being published, so.
Sí. Dr. Lanas.
Professor Barbacid, thank you very much for being here with us and accepting our invitation to participate in the Congress. Well, I'm a gastroenterologist, and I see, as you say, a clear and, I will say, worried increase in the number of pancreatic cancer we are seeing. Almost every week we have young people, I mean 50, 60 years, you know, I'm 68, so it's for me they are young, admitted because of pancreatic cancer. And yes, this is a huge problem. You said the resistance, but we know that the pancreatic cancer, I mean, yeah, in general, everything that affects the pancreas induces a lot of fibrosis. Could it be a problem of penetrance of the drug? During this Congress, we've seen a lot of comments and talks about microrobots, about nanoparticles. Could be possible for these nanoparticles with the drugs that you are treating just being injected in the blood and to increase the capacity of reaching the cancer cells?
Well, thank you for your question. It's a very, very pertinent question. In fact, that's why I said my last commentary that my oncologist friends tell me that their tumors are bigger than my mice. What I mean is that in a bigger tumor, things are going to be much more complicated. Clearly, new technologies with nanoparticle, anything that penetrates the stroma, okay, will be fantastic. Anything that eliminates the stroma will even be better. We, in fact, doing some experiments where genetically we eliminate the hyaluronic acid and we make the tumors much more penetrant. The only problem is we don't have any drug to eliminate the hyaluronic acid in the clinic yet. But anyway, what we do is first we do genetics, and then if it works, we try to find drugs. So clearly the penetration, the size of the tumor, and the role of the dermoplastic stroma, these are things that are going to complicate matters a lot. But at least we need to be a step by a step, okay?
Last question, please.
Translating into clinical trials, I was thinking that this, for the very first time, is treated, I mean, is to say in people that has been already treated with chemotherapy, for example, you will expect different response, or I rephrase the question. Do you think that if we pre-treat patients with chemotherapy, the result that you see in your models will be impaired, or you know?
I have really no way of knowing. Unfortunately, as you know, there is very few chemotherapy drugs. Yeah. There is FOLFIRINOX, which only few people resist the treatment. To be honest with you, I have no idea. I mean, we are not at this level. We can do experiments in mice, but in mice, for instance, gemcitabine works pretty well, and then it's a kind of a false positive. So. I don't know. The short answer is we don't know.
Okay. Thank you very much for the talk.
As you know, the world is divided into those with good hands and those who have a smart mind. Our next speaker, Dr. Shapira, is a rare creature that has both skillful hands and great and smart mind. She was 6 times on the rector list, She is a giftful scientist which paved the way for improvement and breakthrough in medicine. She's just was the awardee of the Falling Wall Award in Berlin, and she serves as a head of the prevention sections at the ESMO, and we are very pleased to tell us about her solutions for cancer. Cracking the code, revolution the pan-cancer screening with a single blood test. Dr. Shapira.
Wow, what an introduction. So first of all, yes, I want to sincerely thanks to Angel and Nadir for this great opportunity to be part of this meeting, and especially in this closing section. And in this presentation, I'm going to describe a potential use of CD24 as a biomarker for early detection of cancer. Now, unfortunately, cancer remains a devastating disease, causing pain and suffering wherever it appears. It kills about 1 in 6 people worldwide, emphasizing the urgent need for new advancements and improvements in cancer prevention, early detection, and treatment. But let's start with some facts and figures. So in 2022, approximately 20 million new cancer cases were diagnosed worldwide, led to 10 million deaths. Unfortunately, it keeps rising. The global burden by 2050 is expected to reach 35 million cases annually. It is 77% increase compared to 2022. And the global cancer death will reach approximately 17 million cases, representing 90% increase. Now, this continuing rise in cancer cases is in part driven by the growth and the aging of the population, but it also influenced by changes in our lifestyles. And to date, prevention efforts have not been able to stop or reverse these trends on an overall level. But there is some good news. 2/3 of cancer death can be prevented, but we need to stick to 4 things. Engage with regular physical activity, eat healthy diet, not smoke, and try to avoid becoming obese. But unfortunately, less than 10% of the people actually live their lives that way. So we understand that the cancer miracle is actually not its cure, but its prevention, or at least early detection. More than half of all new cancer cases can either be prevented or detected early through effective screening to detect cancers at an early stage, even before symptoms appear. Now, it ultimately leads to increased treatment outcomes, but also reduces the mortality and minimizes the need for invasive procedure. So catching cancers early can significantly improve survival rates on a scale that is very hard to achieve through drugs alone. But for that purpose, we need a test. And the ideal screening test should meet several key criteria. It should detect asymptomatic individuals at an early and treatable stage, to be cost-effective because too expensive tests will limit its impact on the public population. It should carry minimal or no risk to the individual. It must be scalable, accessible, and acceptable by the entire population. It should provide consistent and reliable results across different populations and settings. And in order to minimize false negative and misdiagnoses, high sensitivity and specificity are required. But most importantly, as Nadir always says, the test must be simple, non-invasive, not cause discomfort, like a blood test, a liquid biopsy. So I asked one of the AI system software how liquid biopsy technology might evolve in 20 years. And it described it as a seamless integration of handheld devices, holographic displays, and AI-driven healthcare in a futuristic clinic. Now, obviously, we are not there yet. And I'm not even sure that it can be that way. But still, the landscape in liquid biopsy technology in 2044 is likely to be much more advanced than it is today. And the gaps between today and 20 years from now represent not only technological advances, but also a paradigm shift in how healthcare is delivered, moving from reactive treatment to proactive, data-driven care. Instead of treating diseases as they emerge, we need to prevent disease or address the root causes before they even manifest. And the gaps between today and the gap that should be addresses, actually all the challenges and issues that we are currently facing in with the liquid biopsy area, such as low sensitivity, lack of standardization, high cost, and limited accessibility, most of them have already been resolved by the pan-cancer screening test that we have developed, the CancerEnd 24. And the CancerEnd is based, among other things, on evaluating the expression levels of CD24, a tiny molecule that have been extensively studied by our group in the last 3 decades. CD24 plays an important and significant role in many biological processes and has gained a significant attention in cancer research and diagnosis. It is highly expressed in many human malignancies, but also, as we have shown, already at the early and the pre-malignant stage of the disease, while it does almost not appear in the vast majority of the normal tissues. So at the beginning, we focused on pre-malignant and malignant colonic lesions, and we evaluated the expression levels of CD24 in peripheral blood leukocytes. Why them? Because peripheral blood leukocytes are the body sensors. They travel and navigating through the body, entering and exiting tissues with inflammation and neoplasia. Therefore, they can sense and serve as messengers. So data from 150 subjects was analyzed. 63 had colorectal cancer, 19 adenoma, and 68 showed normal colonoscopy. And the results of this study actually demonstrates that the expression levels of CD24, as measured here by Western blot analysis, was efficiently discriminate between the healthy individuals and patients with colorectal cancer or adenoma. Now, interestingly, these differences were not dependent in subject age, gender, size of adenoma, or stage of disease. Therefore, CD24 was suggested as a useful clinical tool for screening asymptomatic individuals for colorectal cancer. Now, these results, this starting point, encourage us to further improve the assay, the methods used, expand the ability of the test to detect other types of cancers as well. And indeed, the cancer was optimized, and it was done in 3 stages. In each one of them, studies were prospective and double-blind. But a few slides ago, I mentioned the lack of standardization in the liquid biopsy field that must be bridged. Therefore, the first thing we did was to ensure that our facility complies with a set of standards and guidelines that were designed to guarantee the reliability and the quality of the data. For that purpose, our work habits have been changed, and countless of documents, procedures, SOPs, and protocols have been written. It ultimately led to recognition by the Israel Laboratory Accreditation Authority as a facility that complies with the OECD principles of good laboratory practice and the United States GLP regulation. Then, once we received the GLP accreditation, we were ready to proceed. And in the first stage, we aimed to evaluate the ability of the CancerEnd to differentiate between cancer and non-cancer patients, including 25 different types of cancers. Now, data for more than 1,000 eligible participants were used, divided to healthy volunteers, cancer patients, and subjects with pre-malignant colonic lesions. And the results of this expanded study actually confirmed that our screening test is able to identify normal individuals versus the cancer patients. And the graph here demonstrated the differences in CD24 score between these 2 groups. However, upon a closer examination of the healthy population, we noticed that the CD24 score among individuals with family history of cancer was slightly higher than those without family history, suggesting that the test may identify family members who have an increased risk for cancer. Then the cancer group has been divided according to the type of cancer, and a cutoff value of 25 was set. Now we can see here some representative results, again showing the power of the test to detect cancer patients while precisely identify the healthy individuals. In a similar way, it was done for other types of cancers as well, but in case of bladder cancer, the test failed to identify those patients. However, when these patients were classified according to the severity of the disease, the test successfully identified those with high-grade malignancy. And when we focused again on pre-malignant colonic lesions, the CD24 score was highly expressed in subjects with polyps, again supporting the previous data that we have shown. When all cancers are classified based on their stage, we saw that the CD24 expression increases early at the disease progression but remains relatively constant regardless the stage of disease or whether it's localized or metastatic. Again, suggesting the use of CD24 as a biomarker for early detection of cancer. Importantly, the results were not affected by fasting or by the time the blood sample was taken, and when it was repeated many times, similar results were consistently observed with relatively high sensitivity and specificity. So based on the results obtained in the first stage, an algorithm has been developed and cross-validated. It represents the CD24 and some other key variables. And for the purpose of developing this algorithm, a cohort of more than 2,000 eligible participants was used. The cancer patients have been classified into 19 major cancer groups. Almost half of them have newly diagnosed cancer, meaning they were recruited before any medical intervention. And since we always like challenges and we always aim high, we have set a statistical goal to find a model that can reach an AUC that is higher than 0.9. And indeed, a univariate logistic regression model was fitted to CD24 epidemiological and clinical characteristics. Now, I cannot expose either the algorithm or any of its characteristics because of reasons of IP. So let's keep it as a black box, or let's keep it a top secret. So raw curve analyses have been carried out for each type of cancer, again emphasizing exceptional precision across various cancers to efficiently detect cancer patients within the healthy population while accurately identify the healthy individuals. And at the end, at the final stage, internal validation has been carried out. It was done in 2 stages. In the first one, again, with focus on colonic lesions, premalignant and malignant, and a total of 652 subjects was analyzed, dividing into 4 major groups: healthy individuals, colorectal cancer patients, and subjects with either adenomatous polyps or hypoplastic one. Then the last 2 groups have been subdivided according to the stage to the size of the polyps. And the results show that the test indeed efficiently discriminates between healthy individuals and colorectal cancer patients, but also patients with premalignancies. Now, classification of the polyps based on their type show a statistically significant differences between the healthy individuals and subjects with adenomatous polyp, but not with hyperplastic one, again suggesting and emphasizing the non-malignant nature of the polyps. And indeed, hyperplastic polyps do not pose a direct risk for cancer. Then further analysis highlighted the size-dependent differences, again, between the healthy population and subject with adenomatous polyps that is larger than 5 millimeters. Then looking at the gender, the same differences have been shown even when each one of them, male and female, were analyzed separately. However, no differences have been shown between the genders in each of the tested categories. And at the end, when the validation was expanded to all types of cancers, the results confirmed that the CancerN is indeed reliable and accurate tool. Actually, it supported potential for widespread clinical application. And this is exactly what we are currently doing. We are evaluating the performance of the CancerN in routine among healthy individuals attending to the Integrated Cancer Prevention Center that Nadir just presented for screening. And this screening test aimed to recruit 2,000 consecutive healthy volunteers, including both male and female, with the aim to show that the CancerN indeed is a supportive tool that can aid physicians with their decision-making process. So I will summarize. We have a game changer. The CancerN is designed to be used in routine medical care to assist clinicians in the early detection of many types of cancers. This is the first blood test ever to detect premalignant lesions. It is compliant with GLP, reliable test, easy and simple to perform with high sensitivity and specificity, simple and fast, and most importantly, it's relatively low cost. So thank you for listening. Okay.
Yes, please.
Lovely story, very exciting data. Look forward to see how it performs, right, in the clinical trials. But I was just wondering about the underlying biology. Do you have an idea how this may play a role in the initiation or progression of the tumors in the colon or other sites that you've been examining? Whether it could be exploited therapeutically, maybe?
We know that CD24 actually accelerates the proliferation of the cells, the migration of the cells. So we suppose that this is one of many pathways that it is involved in. But we don't know actually, but we know that it is highly expressed in the early stages of the disease. We are already showing in colorectal cancer, but in other types of cancers as well. So we know it's connected.
Thank you for the nice talk, very interesting results. So, do you know about the profile of CD24 expression in the blood cells? Because as you may know, CD24 is an inhibitory checkpoint, no? And some new immunotherapy approaches are dealing with CAR T cells or by specific T cell engagers targeting CD24. So,
it is especially expressed on the large cells, especially NK cells, granulocytes, all the population there. And mostly upregulated in cancer cases, but not in the normal state.
okay. Yeah. Thank you.
But on immune cells, yes.
Okay, thank you. And finally, our third speaker will be Professor Marta Trapero. She is a health economist from Lleida. She has a master's degree in the health policy, planning and financing from the London School of Economics. And she has a relevant expertise in the reimbursement of drugs in cancer. She will deal with the topic balancing innovation and sustainability, financing tomorrow's cancer treatments. Please.
Well, thank you very much, first of all, to the organization to have the valency to bring to this absolutely great Congress an economist, yeah? Because probably I'm the last one, but the only one at the same time, probably. Okay, so yeah, I'm the last one, and I will try not to be a party popper, you know, but trying to bring to you some kind of reality to all this, you know, health technology innovation that we have seen, that I'm absolutely astonished to see how we are going to be dealing with this innovation. So I'm very astonished to see all the innovations that is coming, not only in prevention, also in treatment, and all the different kind of, you know, biotechnology progress, the IA, how it's going to be, you know, mixing everything. But I'm also worried, I have to say. And I will explain why I'm worried. Because at the end, although I am an economist, I will try, or one of my points in my research is to try to facilitate access to the population, yeah, to patients, to citizens, to all of us. And I don't really know how we are going to be doing this because we have scarce resources, as you know. Carmen Laplaza was, I think that she's in here, yeah. She was pointing a little bit of worrying also this morning. She was implying that there are some inefficiencies in all countries, yeah, in terms of, you know, the health resources, how we use, she was talking about the use of health resources. And it's not only the wasting or the inefficiencies that we have, it's how we are going to be addressing all this. So I'm going to try to raise like 4 points, yeah? my first point will be why are cancer treatments cost increasing exponentially, yeah? And then I will try to address or just talk about the challenges of the current financial models that we are using. Then I will try to explain or just do some points, yeah? Why or how industry responses and public policy response to all these problems with cancer, and if the intervention that they are doing is sufficient, really, to facilitate this access on the treatments. And we can talk about treatments and also about prevention through the test. And what we can do to ensure equitable access to these treatments. Because the goal really is obviously discuss different solutions to make accessible all these treatments and also financial sustainable. Because there's not going to be access. I mean, we can, obviously that it's an important moment because we are plenty of innovation, we are progressing and all this, but at the end, we need to bring all, to the real world and how we are going to be, assessing this and how we are going to be facilitating all this and using this for the population. At least this is one of my concerns because I've been assessing the Ministry of Health in Spain about the access of drugs. So about raising costs, why exponentially in this disease, we have been dealing with rising costs. Not only with this disease, we have other diseases, yeah, that the innovation have been kind of rising costs incrementally or exponentially. Well, in here, we have groundbreaking innovations. We have CAR T-cell therapies that it's using patients' own cells. We have personalized medicine, which is, tailoring the treatments, which is great, but this implies, some cost and some use of resources. And then the artificial intelligence, which is really accelerating the development of new treatments, and also, trying to progress in reducing the uncertainty, because this is also important. We have been, or at least in the sessions that I've been, we have been listening a lot about the value, yeah, or trying to figure out about the efficacy of the drugs, the treatments, the prevention trends, and so on. But there is always, a huge uncertainty attached to these treatments. And this also, when we need to kind of facilitate the access in the different countries, the uncertainty needs also to be linked to the decision of the price and reimbursement. So this is also important. There is, this disease, and there are a lot of there are very high R&D investment costs in here, and to develop the pipelines really takes quite a long of years. Yeah, so this is really promoting that these costs are really are quite high. Then the economic burden of cancer are not only the direct cost. Yeah, so we have the treatments, but we also have, although are lower than the treatment. At least so far, we have other like surgical costs, hospitalizations, and then there are other costs, the indirect costs, yeah, loss of productivity and financial burden for families that we need to kind of take in account. So we have different case studies of expensive cancer therapies. We have examples, but the dilemma in here is to kind of, balance the affordability with the efficacy of the different treatments. So we have traditional payment structures, so public and private insurance coverage, and this has got, limitations in covering high-cost drugs. We also have out-of-pocket payments by patients, but this really pushes families into financial ruin, unless there are, some public health systems behind. And then we have reimbursement mechanisms that are, depending on the regulation, quite slow and complex. Yeah. So the decisions on public and private insurance and reimbursement mechanisms are disconnecting from measuring the additional relevant incremental benefit. And I haven't heard about that so far in here. So we always talk about the efficacy, the value. So what is value? Value is just an additional kind of, benefit to the patients, or we are talking about the relevant incremental benefit. So what we need to decide, so for instance, why should be, in the next years, funding here in Spain or in any other country? Test, prevention, which we all know, I'm a very, first defendant of public health. So prevention, or should we go, to treatments, to some different kind of strategies to treat? What we should do? Because we have, scarce resources. So we need to promote this information too. So obviously we have reimbursement challenges, value-based pricing, what is value, what is the incremental benefit, how we measure this incremental benefit. If we don't measure the incremental additional relevant benefit, there is no way that we can then link the decisions of access to the kind of public reimbursement decisions. And then the role of economic evaluations, the efficiency. So it's okay to talk about cost, and it's okay to talk about benefit, but we need to link both. It's not only about talking if this is expensive or not. It's about balancing the cost, and the value and the clinical relevant benefit that something health technology is adding. So we have sustainability concerns, and this is important. So are they enough, the strategies and the programs that that we have now? Well, in pharmaceutical industry, which are the pharmaceutical industry strategies? Well, justifying the high prices, so they have, high investments, and therefore this implies a high cost. And but they are, companies. So this is, the health, the economic theory just explains that if you are, a company, you need to raise benefits, and then we have risk-sharing agreements and outcome-based pricing models that try to somehow link or introduce the uncertainty to all these strategies. What the governments and regulatory interventions does so far, so price negotiation mechanisms and also some compulsory licensing in here, and then try to somehow do some partnerships between governments and companies to reduce and share the cost, And we can learn about, other diseases like hepatitis C, rare diseases, HIV, And also to listen, because this morning they were saying, so from the European Union, they were just saying that the interest, which is absolutely normal and that we should be happy, are chronic diseases, because we have the inverted pyramid. So it's important to take this in account in order to design, the road ahead. So what we can do to do more equitable or to facilitate the access to the population? Well, one of the things, and there are already some experiences, not in all countries, but in some experiences, innovative payment models. Governments are kind of trying to think about setting a Netflix model, or a kind of, a subscription model where you pay a subscription. So imagine that you have, some a bag of resources and that you need to split and distribute this money. How can we access to all these kind of different services and innovations? So maybe we need to pay like a quote, and we will have, access to some of the programs or the interventions, but what is sure is that we will have to prioritize, So there is a need for prioritization, and we need to think how we are going to prioritize. If we want to incorporate at least half of the things what we have seen in here. Adaptive pricing based on real-world effectiveness, global cooperation for pricing regulation, and then encouraging investment which is cost-effective. If we want to evaluate the cost-effectiveness or the efficiency of all these innovations, we need to measure really the benefit, yeah? So it's not only the value, so the real benefit, so the relevant benefit, and also the cost, yeah? So the future of cancer treatments depend not only on the scientific breakthroughs, which are really important, but also on financial and policy innovations. And the stakeholders need to collaborate. How they need to collaborate? Trying to sit down in the different stakeholders, sit down in a table and try to establish different mechanisms to find different solutions to fund all this innovation. And I will leave it in here. Thank you.
Any questions from the audience? I'd like just to ask a comment from you. I've seen since, let's say, 20 years since Herceptin came into the market, that the prices of these drugs would be a day that could not be affordable by the society. How do you foresee the near future at that point? Will that be a real possibility?
To incorporate all this?
Yes.
For sure that not. So we need to prioritize. The problem is, which is the criteria to prioritize? Should we go for prevention? Which quote of our resources should go to prevention? Because if we want to eliminate the diseases, otherwise, so we can spend, well, it's not really spent, we can invest a lot of resources, yeah, on treatments. That's fantastic, but we are not sorting the problem. So if we invest our money in prevention, we know that at least, yeah, we are reducing a little bit, you know, the problem. But we need to think about how we are going to be incorporating this, because the pipeline, I mean, staying in this, you know, absolutely great Congress, you see how many things are already there. And we want to incorporate all of them. I mean, economies, health economies, we want to incorporate them. But how we are going to be doing that? I mean, we need to prioritize, and we will need to think how to help decision makers, not only national, so also in Europe, to take the decisions forward, because this is a matter for everyone. This is not, you know, a matter of the ones that are taking the decisions. Yeah?
This is exactly why we need you. This is why you're invited. You should promote early detections and prevention. We shouldn't spend expensive, you know, small proportion of the population, but we should implement it to large part of the population. This is why you're invited, and as the last speaker, so we can understand the conclusions of how medicine is going to look like in 20 years from now. Prevention.
Hopefully we will see, but also treatment. Otherwise, you know, we won't have resources to treat people. So we need to balance it, yeah? Otherwise, if we invest all our, you know, all the money in prevention, we are not attending the population. So it's not that easy, yeah? We will invest 100% of the money, yeah, when everything is sorted, when there are no diseases. Then 100%, I agree. Meantime, I don't think that that's the solution, but it's part of the solution, yeah?
Professor Lanas, please, an important announcement.
Okay, friends, we are at the very end of this outstanding Congress. Well, Nadir and I will just close the meeting. But before we close the meeting, we have the Astor Awards. It has been difficult. But we have to choose some of them. So finally, we have decided to select 3 of the 5. I'm very sorry for the 2 that were not selected. Actually, they deserve it so, but at the very end, so we have to select a few of them. we have chosen three. So number one has been bispecific killer cell engager against T and antigen and non-tissue restricted immunotherapy for cancer treatment by Jorge Paul and collaborators. We have not the money. We will get, no, please come here. Okay. Thank you. You don't worry, you will receive it, all right? So this award is from Frontiers in Medicine Gastroenterology. It's a gift of $1,000 that is going to be divided by 3, all right? Okay, the second one is going to be for Andrea Moreno and collaborators with targeted immunotherapy for pediatric neuroblastoma patients. Okay, and ex aequo, the third is going to be for mice, Motlac, the cancer and D24, a novel blood test that can prevent colorectal cancer. Thank you very much. Okay, so I will. Sorry, Nadir told me 500 each, okay, which is much better than, you know, they are generous. You know, Israel is much richer than the Spaniards. Okay, well, before I give the floor to Nadir to close the meeting, I just want to say that I really thank you all for being here, not only those who are till the very end, but everyone who has participated. And especially I want to thank all the collaborators. Many people have been involved in the organization of this meeting. I'm not going to say names because I'm sure I'm going to forget some and then I will be killed tomorrow. But many people have been involved. And also a special thanks for all the for all the presenters, all the speakers, all the chairs. You know, everyone has participated. Everyone has put a lot of in this conference. And finally, I want to thank also all the sponsors. Again, I'm not going to name them, but you have it, all of them right here. So then, without that help, it would be impossible to have celebrated this Congress that I think that for Zaragoza has been something very important. 2 hours ago, I was interviewed by Cadena Ser, which is a radio, a quite famous radio in Spain, and also by the Aragon TV and some other local and national radios. And they told me, okay, Dr. Lanas, what can you say as a summary of this meeting? Can you say this in 2 words? And I say, no, I mean in 2 sentences. I say, no, I just only need 1 word. Extraordinary.
I absolutely agree with Angel. This was the perfect meeting. I think if at 7 o'clock on Friday night there are so many people, this gave that this is a good meeting. I think this is a way to go in order to try to prevent disease rather than treating disease. We have to plan how medicine research are going to look like 20 years from now, and I'm very pleased that we are continuing with this series of 2040 medicine series. It was a perfect place. We like Zaragoza, and especially because the people here are good people. Warm heart, friendly, and I think this is hopefully that we're going to be the entire world can learn. And obviously we are going to continue with these traditions. Hopefully other cities and others will join us and we can spread what actually government should do, how to plan the future, not to target the disease itself. And mostly we should thank Angel. Without him,
Okay, we are very tired, so it's time to go home. Yes.